So, in this video, I would like to explain the various plotting techniques, including southern plotting, northern plotting, and western plotting. Before getting inside the plotting techniques, I would like to recall the general dogma of molecular biology. So, in this case, the DNA can be replicated into another DNA with the help of DNA polymerase. Moreover, this DNA can be transcribed into RNA, and this RNA can be translated into protein. So this is the central dogma of molecular biology. So this can be make reverse in this case of RNA can be again converted into RNA with the help of enzyme reverse transcriptase from rotavirus. So reverse transcriptase enzyme will help the RNA, that means mRNA, into DNA. That is reverse transcription process with the help of reverse transcriptase. So there will be a relation between DNA and RNA and protein. So DNA can be transcribed into RNA can be translated into protein. So the technique which detects DNA is called southern plotting technique. And the technique which detects RNA called northern plot technique. Then protein detection is called western plot technique. So this southern plot technique is discovered by the scientist Edward M. Southern. So it is named after the scientist Southern, Edward M. Southern. So later on, the RNA detection and protein detection are named as northern plot and the western plot for protein detection. So nucleic acid plotting is a well-established technique for locating a gene or a sequence of interest from a complex mixture of DNA or RNA. So southern plotting and northern plotting can able to detect any gene or a sequence of interest from a complex mixture of DNA or RNA. In this case of western plot analysis can detect protein of interest from a mixture of great number of proteins. The plotting refers to the process of immobilization of sample like nucleic acids or proteins on solid support such as nitrocellulose or nylon, etc. So the process of immobilization, that means there will be an attachment of nucleic acids or protein with a solid support. So this can be done by adsorption process or cross-linking process or covalent bond process. So the immobilization process can be takes place by adsorption, cross-linking, covalent bonding. So this immobilization process will make the nucleic acid or protein attached on the solid support. That is called plotting. So in this plotting, nitrocellulose paper is most probably used in the plotting. Nitrocellulose paper is used in the DNA plotting, southern plot. Nylon or diazobenzyl oxymethyl or aminobenzoxymethyl paper are used in the RNA plotting or northern plotting. Nitrocellulose or polyvinylidene difluoride (PVDF) paper is used in the protein detection, in this case of western plot analysis. So this picture showing the common procedure for plotting techniques. So just imagine this is the RNA or DNA or a protein, whatever, right? So this is a common procedure. So in this case, it's stating the DNA, right? So the first step involved in the plotting is the digestion or cutting into small pieces in order to separate the DNA or RNA or protein of interest from the group of fragments. So the longer DNA bundle can be cut into small pieces, right, with the help of restriction endonuclease enzyme. So this is the DNA of interest what we want. We are going to detect. This is the DNA of interest. So in this case of Nathan plot, so this can be done with the help of restriction endonuclease along with formaldehyde solution. So the formaldehyde solution will make the RNA fragment linear, which can able to easily separated with the electrophoresis. So the first step is cutting into smaller fragments, either DNA or RNA, and in this case the western part is a protein can be denatured with the help of SDS, sodium diacetyl sulfate. After the first process is over, this DNA or RNA or proteins are separated using the electrophoresis technique. So electrophoresis is the second process in the blotting technique. So after the electrophoresis is over, then the third process starts, that is the transfer of the gel containing protein or RNA or DNA into the membrane. So transfer of DNA or RNA or protein into the membrane, that is the transfer. Then the fourth step is that hybridization. So here is the important principle involved in the plotting techniques. So in this case of hybridization or detection involves the binding of specific probe into the particular DNA or RNA or protein of interest. So in this case of DNA detection, that means southern plot, DNA hybrid will be found. With the help of DNA probe. In this case of RNA also the same thing will happen. Here RNA DNA hybrid will be found. In this case of protein detection, western plot, instead of hybridization, antigen antibody reaction will be takes place. I will explain later about it. So fourth step is the attachment or detection. So after detection, there will be a autoradiography. That means development of film or identification of particular DNA or RNA of interest. That means autoradiography. We may call in other names gel documentation system. So in the entire process of plotting techniques, that includes digestion, cutting into small fragments, then electrophoresis, then transfer into membrane, then detection, then identification. So these are the common steps involved in the plotting techniques. So in this case of electrophoresis, so electrophoresis is the technique which makes the DNA or RNA or protein will get separated. The separation can be done with the help of gel. So normally southern plots and northern plot will utilize the agarose gel. In this case of protein detection, western plot will use the polyacrylamide gel. So in this case, commonly we are using the chamber like this. This is a rubber jacket. So there is a space for pouring the gel. So normally the agarose powder will be mixed with water and dissolved with high temperature. When the powder will become a liquid, it will be poured inside this chamber, inside this rubber basket. Once it is poured in a liquid condition, this comb should be immersed in the liquid, this comb, which makes the well or hole after solidification of this agarose. Once this liquid agarose get solidified, right, this comb will be removed, which makes the formation of well or hole. In this well or hole, we will load the DNA or RNA or protein samples. Protein samples. Usually, the first hole will be filled with the marker, either DNA or RNA or protein markers. Protein markers. In other words, we may say ladder DNA. Ladder DNA or marker, which is used to compare the DNA or RNA or protein according to the molecular weight, according to the size or molecular weight. So once the DNA or RNA or protein samples were loaded in the well, right, we may pour the puffers inside the tank, right. After the puffers is poured, we may switch on the electric current. When the switch on the electric current, the movement of molecules will start. So the DNA or RNA or protein will move from negative electrode to positive electrode. Negative electrode to positive electrode according to the size. According to the size. In this case, larger DNA or RNA or protein fragment will move slow. In this case of smaller DNA or RNA or protein fragment will migrate or diffuse faster. So smaller fragments will diffuse faster than the larger fragments. So the movement will occur from negative to positive electrode, since the DNA and RNA are negatively charged. So these negatively charged molecules used to move towards the positively charged electrode. Once the separation, the movement is finished, then this gel is taken away from the upper system. Then we can go for the transfer of gel containing DNA or RNA or protein band into the membrane. That means either nylon or nitrocellulose or any other membrane paper, right? So this is the system used in the membrane transfer. So this is the plastic tray containing buffer solution. So this is the filter paper wick. This is the supporting block, right? In this, after keeping some filter paper wick, we may keep the gel first. The agrogel or polyacrylamide gel, we will keep it first. Then above the gel, we may keep the membrane. Then above the membrane, we may keep some 3 sheets of filter paper. Then above the filter paper, we may keep some paper tissues. Then we may keep glass plate. Above the glass plate, we have to keep some weight. So after keeping it, the buffer solution will start move from high water potential to low water potential. So during the buffer movement, with the capillary action or capillary transfer, the gel containing DNA molecules move from gel to the membrane through the capillary action. With the help of capillary transfer, the DNA gel containing DNA or RNA or protein band from the gel will move and go and attach with the membrane, with the nitrocellulose membrane. Since the DNA or RNA are negatively charged and the membrane mostly it is a positively charged, the movement will go and attach with the membrane. So this transfer is occur through capillary transfer. In this case of DNA and RNA detection, it will occurs mostly through capillary transfer. But in this case of protein transfer, we may use the electric transfer also. We may use the electric current for movement of protein from negative charged to positively charged electrode. This transfer may take place very faster when compared to the capillary transfer. So electric transfer may happen faster than the capillary transfer. So after transfer is done, then there is a attachment or immobilization is done, there will be an attachment or detection of DNA or RNA or protein. So this figure showing the comparative principles of plotting techniques. So this slide showing the comparing the principles of plotting techniques such as southern plotting, northern plotting and western plotting. So first one shows the western plot. So in this case DNA sample is mixed with restriction endonuclease, cut it into small fragment, then we are going into the agarogel electrophoresis. After separation we are transferring into the nylon paper or nitrocellulose paper. In that there will be a DNA. It is a single stranded DNA, right? In that stage, we are adding the DNA probe. DNA probe. This DNA probe is a single stranded DNA piece or single stranded DNA fragment. Single stranded DNA fragment having the sequence complementary to the particular DNA of interest. So this DNA probe, which is used for hybridization, must have the DNA sequence which is complementary to the particular DNA of interest what we are going to detect. So we are using the DNA probe which is having the sequence which is complementary to the particular DNA of interest. So when you add the DNA probe labeled with radioisotopes or labeled with some fluorescent material, it will go and pair with the sequence. For example, the probe containing cytosine will have a base pair with a guanine. Then the thymine containing probe will base pair with the adenine. This adenine of DNA will base pair with the thymine of DNA probe. So accordingly, this DNA probe will attach or hybridize with the particular DNA of interest. So this DNA probe will attach or hybridize only with the particular DNA of interest among the group of DNA. So this is the basic principle involved in the southern plotting techniques. So how we detect the DNA of interest means by using the single stranded DNA which is having the sequence which is complementary to the particular DNA of interest. When you go into the northern plot, the same thing will happen. So here we are using formaldehyde, then agarogel electrophoresis separation, then transfer, then there is a detection with the help of the same, like here also we are using the single stranded DNA probe which is labeled with radioisotope or some fluorescent material or any dye. So this probe is complementary to the mRNA. So this DNA probe containing sequence will attach or paired with particular nucleic acid bases. The same thing happened in this case of northern plot as southern plotting technique. So northern plot also the base pair will happen, hybridization is occur. So the hybridization product is RNA DNA hybrid. In this case of northern plot, DNA hybrid. So southern plot we have a hybridization product of DNA hybrid. In northern plot, RNA DNA hybrid will happen. When you go into the western plot, we are using sodium tetraethyl sulfate for denaturation of the protein, followed by polyacrylamide gel electrophoresis for separation. Right? After separation, it is transferred into the membrane, nylon or nitrocellulose membrane. So after transfer, the detection is done. So detection is done in western plot based on antigen antibody reaction or antigen antibody complex formation. So you know the proteins are the antigens, right? Proteins are the antigens, right? Or antigens are the proteins. So we are going to use the antibodies that is specific to the particular DNA of interest, protein of interest. So we are going to use the antibodies which is specific or particular to the protein of interest, what we are going to detect. So in this case, we are adding antibodies, primary antibodies. First, this antibodies will go and bind with particular antigen specifically due to the affinity between the two. So due to the affinity of this antibody towards the antigen, particular antigen, the primary antibody will go and bind with the particular protein of interest. It will make a complex. It will make a complex. Then we are adding the secondary antibody labeled with fluorescence or any radioisotopes. This secondary antibody will go and attach with the primary antibody. So there will be a complex of antigen, primary antibody, secondary antibody conjugate. Secondary antibody conjugate. So that will makes the detection of particular protein. So Western blot analysis working in the basis of antigen antibody complex. Antigen antibody complex. So after this detection is over, we can able to visualize the particular DNA or RNA or protein with the help of autoradiography. Autoradiography. In case if you attach the DNA probe with the radioisotopes, we can go with X-ray film. X-ray film. If you attach with some fluorescent material, attached with or labeled with fluorescent material, we can go with chemiluminescence. We can go with chemiluminescence. If you attach simply with some dye, you can go with some colorimetric analysis. Colorimetric analysis for detection. Colorimetric analysis for detection. So this slide showing the comparison between south, northern and western blot analysis. So now we may compare the entire process and principles of three plotting techniques. So first one showing the southern plot, second one northern plot, and the final one western plot. So in this case of southern plot, it will detect the DNA molecules. So southern plot techniques will detect the DNA molecules. Northern blot technique detect the RNA molecules. Western blot detect protein molecules. Western blot detect protein molecules. In southern blot, we are using restriction the nuclease enzyme for cutting and digestion of lengthy DNA fragment into small fragment. Here we are using formaldehyde for making linear RNA, linear mRNA molecule. In this case, a western blot, we are using sodium diacetyl sulfate for denaturation of the particular protein sample, Followed by next is the agrogel electrophoresis separation. In the separation, both southern and northern blot will use agrogel electrophoresis, but western blot will use SDS beige, sodium diacetyl sulfate, polyacrylamide gel electrophoresis. So in this case of DNA or probe, southern blot and northern blot will utilize single standard DNA probe. So both nucleic acid blot will use the single standard DNA as a probe for hybridization. But in this case of western blot will use antibodies. Primary and secondary antibody. Secondary antibody is labeled with fluorescent or any radioisotopes. So the product of identification is based on DNA hybridization in southern blot. In northern blot, RNA-DNA hybridization. So in nucleic acid plotting techniques, southern blot will have a DNA hybrid. In northern blot, RNA-DNA hybrid. But in this case of western blot, antigen-antibody complex. The paper used in the different plotting techniques. In this case, mostly southern blot, we are using nitrocellulose. In northern blot, we are using nylon or diazobenzyl oxymethyl paper or amino benzoxymethyl paper in northern blot technique. In western blot, we may use nitrocellulose or PVDF membrane. So in this case of mechanism of transfer of DNA or RNA or protein from gel to the membrane will occur through capillary transfer in southern and northern plots, and also western plot. Only the western plot, the electric transfer also possible in the western plotting techniques.